I wanted to welcome everybody to this week's Research and Discovery Seminar Series. This talk, we have as our speaker is Dr. Sean Mooney, who's actually our Chief Research Information Officer. So he's high up in the executive team at UW Medicine. And he's also the interim director for the UW Institute for Medical Data Science, as well as a professor in the Department of Biomedical Informatics and Medical Education. So Sean got his start, got his PhD at UCSF in pharmaceutical chemistry, and then he moved on to just down the road to Stanford University, where he was an American Cancer Society John Peter Hoffman Fellow for a couple years, and then took a faculty position in Indiana University School of Medicine, where he rose through the ranks, was the founding member the founding director of the School of Medicine Bioinformatics Core, as well as a member of their Computational Biology and Bioinformatics Center there. And then after that, he moved on to the Buck Institute, where he was there for a number of years before landing here I believe in 2015. And he's been here ever since. And his main focus, as you can imagine, is in biomedical informatics. And he has done a huge amount of work establishing and setting up the processes to use medical informatics in healthcare. And I'm continually asked, because of my experience in sequencing, on what kind of infrastructure and how do I do this and how do I do that, and so a lot of those questions I don't know the answer to, and I don't even know what's possible. And so I thought, why don't we hear this from the person who probably is one of the most informed at the university? And so with that, we look forward to hearing what you have to say.
Awesome. Thank you so much for the kind words and the introduction. I really want to thank the organizers of this seminar for inviting me to give a presentation. I love giving talks, and I particularly love giving talks to a University of Washington community. This is going to be an interesting seminar for me. This is the research seminar, so I'm going to kind of dabble between operational activities that we do and more researchy sort of projects that my research group has done. And so some parts of this will be a little bit eclectic, but hopefully you'll find it interesting. And I'm hoping that at the end of the day, you learn a little bit more about how, at UW Medicine, with all of the electronic systems that we've put in place, like our EHR and our enterprise data warehouse, and computing infrastructure, that we actually have the ability today to not only think about, developing machine learning or artificial intelligence methods based on data, but actually implementing them and piloting them as part of a QI project or as part of a pragmatic clinical study. So what I'm going to do is I'm going to start with a story, and then I'll move into talking a little bit more about the infrastructure that we have here in place, and hopefully connect maybe some of my career. I started as a chemist and then did a lot of bioinformatics from a genomics, or a protein chemistry perspective. And I kind of want to maybe tell a little bit about that story about how I got here, and in a place where really we're trying to leverage our rich data resources at UW Medicine to enable more data-driven improvement in how we provide care and how we run our business. I also should say I've got a lot of slides, so I may not get through everything. If I don't, I'm just going to skip things and just go to the end. I hope that's okay. Happy to share my slides at the end of the day if I miss anything. So I'm going to start and talk a little bit at the highest level. Computers are excellent problem solvers. We know this. In 2007, checkers was solved completely by a computer. That means the computer could enumerate every possible position in a game, and you could not beat it. Because every move, the computer could then say, what are all the possible future moves, and solve that. And so checkers became a solved problem. In chess is more complicated, but chess has repeatedly beaten humans, the best humans. And in fact, a pocket phone device was able to beat a master level chess player in 2009. And chat right now at this point, also chess is computer systems that run chess are extremely good. And then I'll mention, we've all probably seen ChatGPT. I really do think it's a stunning advancement, and I think it's just opening the door to a whole new area that has a lot of opportunity and a lot of uncertainty in terms of what that actually means for for us in biomedicine, and also us in a university as professors who teach students. So let's step back and just kind of look at some very high level things. Today, there's 5,000 to 8,000 known diagnosable rare diseases. That's more diseases or conditions than any individual provider could diagnose on their own. It's estimated somewhere between 25 and 30 million Americans have a rare condition. I've seen that's estimated about 1% of the population. I've seen estimates as high as 7% of the population, so that number could be sevenfold higher than that. And in fact, there may be lots of individuals who are walking around with rare conditions and don't know it. Most rare conditions have a genetic component, and it's an important problem, many of you know, is identifying causative genetic elements. We call them pathogenic variants, and we would like to identify them from benign variants, or that is, genetic changes on the human genome that don't cause disease. We've passed 20 years since the initial draft of the human genome. It had much fanfare. It became a platform by which we work on. A number of high throughput sequencing technologies have become commonplace and have been put into place that enable, high throughput sequencing of human genomes at extremely low cost. And the analysis tools are becoming, much more easy to use and manageable. And we can identify, essentially the library of genetic changes or genetic variation in any individual. And we can use that to characterize ancestry. We can use that to characterize risk for diseases. We can identify causative pathogenic variants, like I mentioned, on the human genome, to identify risk of, say, children in a genetic, for a genetic counselor who's counseling prospective parents. Typically, the way that we identify those variants, the variation that is disease associated, is we do it through association. We basically take groups of individuals that have something like a condition or a disease, and a group of individuals that don't, and we look at genetic variants that seem to propagate statistically toward one side or the other. It's a very statistical approach, and those statistical elements that we identify are those statistical elements may or may not be actually causative of the disease. They're just simply associated. And that is, if we interrogate that in other people that were not part of that study, we may be able to identify whether they're at risk for that condition, or maybe even have that condition based on the presence of those variants. But if we were actually to take, like, say, a rare disease, and we were to identify a pathogenic genetic variant, and we were to then look at what that variant is expressing as and doing on a molecular level, we'll find that pathogenic variants, maybe not surprisingly, affect biology. So in this case, I'm showing an image of a ribbon diagram of the p53 tumor suppressor protein, which is well known for having amino acid substitutions that are risks for cancer, both inherited and somatic. And we can take mutations that we know about, or pathogenic variants that we know about, map them to that structure, and we can immediately see on a molecular level how they're disrupting the molecular biology of p53 in order to give rise to the condition that p53 is associated with in that patient. So we've developed machine learning models. This is something I've been doing in my career for almost 30 years now, 25 years. And we build machine learning models that basically will take a genetic variant that we've never seen before, or a genetic variant we have seen before, we don't know what it does, and we try to predict whether that variant is a pathogenic variant or a benign variant. And the way that we do this is very simple. We use supervised machine learning. We have a variant that we've, again, that we've never seen before. We generate a list of features or attributes that describe that variant based on biology. And then we ask the question, does it look more like a database of known benign mutations or known benign variants and a database of pathogenic that cause disease? And if it looks more like the pathogenic ones, then we will, be more confident in predicting that that's a pathogenic variant. All right, so many of you are probably familiar with ROC curves. As I walk through this presentation today, I'm going to talk about ROC curves quite a bit. So I'm going to show you one here. Receiver operator characteristics plots are really awesome. They're a very easy way of measuring how accurate a predictive, a machine learning model that's making predictions is doing. And they were originally developed by the US military in World War II. They have a fancy name because they were basically the mechanism by which we could measure the accuracy of receiver operators. Those were basically people looking at radar information and trying to make a determination of whether an aircraft that was approaching the radar was enemy or friendly. And they developed this metric to be able to determine or make a determination about whether, how accurate the whole system was. And it was extremely effective at being able to compare different things. Like you could swap out technology, you could swap out the person, et cetera. And the way that ROC curves work is that if it's a perfect prediction, that means it always gets it right. You'll see a curve that looks like the A curve that's got an arrow there. If it's sort of in the middle and it gets some things right and some things wrong, it'll look like the B curve. And if it's completely random, like a coin flip, it'll look like the C line through the middle. And if it's actually getting things backwards, it'll be a curve that's not shown here that would be underneath the C line. I love ROC curves because the area underneath these curves that fill up the box underneath the curve actually has statistical meaning. And it's basically, if you were to rank all of your predictions, rank all of your predictions from the top prediction to the bottom prediction by your method, and you were to take a random known positive case, like in the case of a disease, the random individual that has the disease and a random individual that doesn't have the disease, the area underneath the ROC curve is the probability that you've ranked the case above the control. So we've developed a method called MutePred2. It's published, it's very widely used, and what it does is it looks at, in this case, protein mutations. Those are amino acid substitutions in proteins. MutePred is a suite of tools, so we have also versions of this that look at other types of variants, both in genes and outside of genes. But I'm just talking about the protein one here, and that's probably the one that we're most known for. And basically what we do is we take a mutation site on an amino acid sequence. We develop a bunch of features based on, basically bioinformatics, and we then use those features as input into neural networks. Basically, what we're interested in doing, and I think this is somewhat provocative, is what we're trying to do is build a bioinformatic model of biology, and then to measure basically measure how a mutation disrupts that biology when it's present, using these sort of machine learning methods. So if you look at the bottom right-hand corner of the features that I've got listed here, we have a long list of functional changes. That is, things that can happen, things that are functional in a protein that would happen, and we can then use those features to make predictions about whether a potential pathogenic variant is actually disrupting those features and then associated with disease. MutePred is widely used. MutePred2 is widely used. The features are also used in another widely used consensus method that was developed at Stanford by ClinGen called Revel, that you may have also seen. And so we sort of influenced both of them. We've published this. MutePred2 was developed by Vikas Pajevar, who's now an assistant professor at Mount Sinai, and will give you both a score that predicts how likely it is that your variant that you're looking at is pathogenic. It will also give you a hypothesis about what function is being broken in order to get you to that pathogenicity, and that score can then be used to compare to others. We've used this, so this is the first set of ROC curves. We've used MutPred2 against a bunch of widely other used methods, and we've shown that MutPred2 is more accurate than the other methods when applied to an independent data set that we did not use in the construction of our method. And so again, we think that MutPred2 works pretty well. Recently, I'm not going to talk too much about this, but Vikas, while he was in my group, and then when he moved on to Mount Sinai, has been leading a committee as part of ClinGen to look at calibrating these computational in silico methods for pathogenicity prediction. And I think we can make an argument now that over the last decade or so, we've had a breakthrough about these methods, and these methods can now you annotate some variants with high confidence and high evidence that they're either likely pathogenic or likely benign. And I think this is really an important advance for these types of methods, because I think over time, you're going to see them improve more and more. One of the things as I start transitioning to talking more about UW medicine that I really want to emphasize is that we need to move beyond an ROC curve. So I just told you that we took a bunch of data, we then built a bunch of features that describe that data, and then did machine learning to try to predict something clinical that could be used clinically, potentially used clinically. And then we published a paper with a bunch of ROC curves where we independently assessed our method compared to other methods. And of course, I told you that our method was the best. And one of the prime things that I want to make an argument for here today is that I want to, suggest that we need to move away from ROC curves and move more into trying to understand how does a machine learning or an AI-driven method change with its actual use clinically or whatever its application may be. The current academic model, as I said before, is to develop a data set, to develop a method, to assess the method using something like cross-validation, and then to test it on a validation data set, and then write a paper, maybe comparing it to other methods. And that doesn't tell us anything about an actual clinical model. It doesn't tell us what happens if the model is implemented into a clinical workflow. We don't know if providers actually use it. We don't know if they're satisfied with it. We don't know if it distracts them from their care. We don't know if there's patient benefit. We don't know if it shows the value that was proposed clinically. And we don't know if it brings value to the health system. There's a laundry list of questions that I haven't answered by showing that MutPred2 is an accurate method. And so that's really, if you leave anything from this presentation that I want you to kind of understand is that that's where we're headed for. We have to get away from ROC curves and get more toward, understanding what happens when methods are actually used. Because it's very easy to develop a method today that's useful, that looks useful on paper, but it isn't clear whether it would actually be useful in practice. As many of you know, so this year, the University of Washington is going to spend between $7 and $8 billion, I believe. About half of that is treating patients across 4 hospitals, 60 or so primary care clinics, the UW medicine neighborhood clinics, our airlift, airplanes and helicopters, our trauma center, our cancer center, about 400 specialty clinics, our School of Medicine. We see actually nearly 1 million patients a year. And, through that, we have 2,500 faculty, 5,000 trainees, and a EHR system that we put live at the enterprise level. So everybody's using the same piece of software basically to deliver care, which is Epic. And from that, we also have what I think is a real forward-looking enterprise data platform that has 30 years of data and more than 5 million patients in it. So one of the challenges as we think about machine learning is something that's been known for decades, and that is this sort of balance around clinical decision support. So like if we were to start building methods like MutPred2 that help providers make decisions clinically, we have a balance between, getting it accurate and reducing errors, improving care potentially, and potentially even making providers more satisfied, along with the problem of over alerting and alert fatigue. And that is, false positives are very common with machine learning methods, and false positives can take away from the efficacy of the original method that may be quite accurate. So for example, UCSF did a study, there's the paper here, where they looked at ICU patients in 2014 and found that there were 187 audible alarms per bed per day, which certainly is drowning out the signal from basically, literally in this case, all of the noise. And it makes it very difficult to imagine how, even accurate alerting within an ICU setting is going to enable, anything useful at all. All right, at UW Medicine, the last 7 years, me and others have done an enormous amount of thought around what it would take to take a data science, an advanced statistical method or a machine learning method and implement it as part of either a clinical trial or like say a QI project as part of, advancing a learning health system. And I try to annotate these here. I think it'd be awesome to talk to some of you if you're interested in having a conversation about this, because I could be wrong. But, essentially, this is a list of like all of the steps that would be required to take us from, nothing to be able to, like, say, alert a provider because a patient's at risk for some condition based on their data. And we need, to start, high quality data. We need governance for that data. We need mechanisms to access that data. We need computing environments that could be in the cloud for doing HIPAA compliant machine learning or, AI kind of driven analytics. We need that software to deliver healthcare, like the EHR, the lab system or whatever. We then also need the ability to build technical interfaces between those different tools, between the data scientist, and that software. And maybe the computing that runs this method. We need to be compliant with regulatory issues like the FDA. If you start replacing doctor things with computers, the FDA is going to want to have a conversation with you. And we also need to have internal, research and clinical governance. So that is, we need to be able to prioritize as a health system what projects we're going to go after, because we have way more projects than, say, UW Medicine as a health system is going to have the ability to do. We're going to need machine learning governance. Anesthesiologist comes to us with a decision support tool for procedures, for dosing, that's based on patient data. Basically, it helps the anesthesiologist dose a patient during a procedure. And then, I don't know, say I come to the system with MutePred2 and I want to implement it into a workflow that is going to be annotating human exomes that are sequenced clinically. How do we make a decision about whether the anesthesiologist gets resources from IT and data, or I get resources from IT and data as a geneticist? And then we need to be able to implement them, and then we have to have the resources to be able to study continuous improvement, over time, to basically maintain these methods, to ensure that the methods are giving us the accuracy and the efficacy that, and the lack of risk that we expect when we put them into place operationally. I mentioned we have 30 years of data with 5 million patients. We've been working very hard on my team to basically make our data better. We do that by extracting concepts from text. I'll talk about this a little bit in a bit. We want to annotate social determinants of health for our patient population, environmental determinants of health. We want to integrate external data sets such as mortality data from, like, say, the Washington State death records. We want to be able to geocode patients if someone's doing, some sort of GIS analysis of our patient data as a research project. And we want to integrate all the other fancy kind of data sets that might be out there that could be useful for research, like genome sequencing or genomics data that might be matched to a patient, or a specimen that's matched to a patient. And this data set that we use from the UW Medicine EDW is fully identified. It's used operationally, but it is also used for research. And the path to integrating into the healthcare enterprise is extremely short. And I would argue, actually, we're as short as any university in the country. But it would have to get through governance and have the appropriate resources to be able to do that in practice. So one of the first things that we did was we developed a tool called Leaf. Maybe some of you know Leaf. If you don't, you're welcome to send me an email and I can connect you to how to get access to it. Leaf is now an open source web-based tool for browsing clinical data from an EHR. And it's basically a window into our enterprise data warehouse. And it is very easy to basically technically click in to any clinical data set that could be clinical research data set that's patient-centric. And we do that for a number of different projects, both here and nationally. It's HIPAA compliant. It's used both clinically and for research, and can de-identify data sets for compliance in real time while you're doing your browsing against the data. And it is in place at UW Medicine. It's been around for about, I don't know, 4 or 5 years now here. And we've got a paper on it, and you can go to the GitHub and get access to the code, et cetera. If you have a data set that you'd like to click it into. Leaf is now widely used. It's been developed by Nick Dobbins, who's the head engineer on my team. At UW Medicine, I think we have about 450 investigators now who have access to it. And they've done tens of thousands of queries. They can export into our electronic data capture tool called RedCap. It's been so successful here that we've actually, it's actually now being adopted at around 30 other sites around the world. And in fact, I've just mentioned Vikas Pajaver, who took an assistant professor position at Mount Sinai. One of the first things he did was ask for access to clinical data, and they trained him how to use Leaf, which was just really, really awesome and a great moment in my career. But if you'd like to use Leaf, you can use it. The GitHub is just easy to download, and it's all yours. And we can, I can connect you to Nick Dobbins, who can help you get access to it. It's a really nice user-friendly interface. It's kind of like a better I2B2 than I2B2 in terms of the interface. Web-based, allows for basic dashboarding to look at patient populations. You can look at a wide amount of continuous and structured variable data. And again, it can be exported into other formats compliantly, such as RedCap, and has both identified modes, if appropriate, and a de-identified mode for doing research. Appropriate research. Again, widely used. If you'd like a demo of Leaf, we can make that happen for you with Nick and the rest of the folks on the Leaf team. UW Medicine. So Leaf is very good at structured data. That is data that's kind of fits into a relational database. But at UW Medicine, we have well over 100 million pieces of text. I actually think it's probably more like 2 to 200 million or more. These are clinical reports, like radiology reports. They are clinical narrative text, like outpatient notes, for example, discharge notes, et cetera. They are, maybe secure messaging within our system. And they're very hard to do any sort of computing with, but there's a lot of rich data within clinical text that we are aware of that we'd like people to have access to. And so we have developed, working with a faculty member by me, Miliha Yediz again, who is really awesome. And if you are looking for someone to collaborate with around doing a specific project with text, highly recommend reaching out to her. We've set up a pipeline that looks approximately like what I'm showing here, where we can take text that's like, say, a note, and we pass it through basically a set of computational methods that could include machine learning that will then extract things from that text and then either create a database of it or create a new note that's been processed based on that algorithm. And things that we can do today, we can de-identify notes. So any of those 100 billion, any note, clinical narrative text, we can de-identify on the fly and do that as a service for you. So if you want access to de-identified notes, we can do it that way. We can also extract biomedical concepts arbitrarily or clinical findings from text, both in an unsupervised way. So like, give me all the notes that mention the word cancer or mention the concept of cancer, or we could actually train a model based on a known set of patients. So if you have like, say, 50 patients with colon cancer and 50 patients without colon cancer, we can then annotate that and build a new model that can predict, annotate colon cancer from notes based on that training set. And then finally, we have a strong interest in leveraging machine learning to extract social determinants of health, structure them, and input them back into EPIC. Continuing on that vein, Nick Dobbins and a staff member also on my team, Richie Long, have been working with the CMIO's office as well as Miliha to demonstrate that we can extract social determinants of health from various notes and use those extractions, get accurate extractions. And we are looking at structuring them and putting them back into EPIC so that they're more available to providers. And what we found when we first looked at alcohol use, tobacco, drug, employment, and living status, like homelessness. And we found that if you look at the table at the bottom, based on this deep learning approach for extraction, that we find that, like, say, living status, like homelessness, is only structured in EPIC for 64% of the patients that we see. And if we look at note text, we see 32%. So basically 50% or 1/3 of the total population is only found in clinical text that we can extract and is not structured. And so we think that this is a promising approach to vastly improve how we annotate social determinants of health within EPIC and make them available to providers. Something else I'm just going to tease, this is actually Nick Dobbins. In addition to being an engineer, he's also a PhD student, and working with Miliha Yediz again. This is pretty independent of my group, and really his thesis has been developing something that's really cool. And that is, if we take, like, say, the inclusion criteria of a clinical trial, like we want all patients that have a BMI between a certain range in the past 6 months, were treated with some drug, are aged some age range, and have been seen in the emergency department in the past 3 years. What this tool will do, which he's calling Leaf AI, will actually take these criteria and turn that into SQL to do a query against our clinical data, and then identify a cohort based on that in a completely automated way. And has shown that if we took, like, say, clinical trials, clinicaltrials.gov data, inclusion criteria data, he can basically very easily apply this tool and get a long list of cohorts that are eligible for basically all the clinical trials that are available on clinicaltrials.gov. All right, so that's Leaf. Leaf is awesome. I really hope you use it. You can use it to access UW medicine data, but you can also use the software to access any other data set that you want, and you can implement it because it's open source and free to use. We have a culture of open science in my group, and so we like to disseminate and make things available when we develop them. I'm just going to mention synthetic data because I'm probably going to talk about it a little bit more later if I get there. And synthetic data is another advance in the AI machine learning world that I think is something that we should all be aware of. If you go to the website that I put here, thispersondoesnotexist.com, it will show you a picture of a person that doesn't exist. It's basically a synthetic individual that was created based on training from a large database of individuals, of pictures of individuals. And synthetically derived data about people is very likely privacy preserving because this data has been munged to the point that it's not really attached to anybody that was used as part of the training process. And why is synthetic data important to us? We rely a lot from a compliance perspective when we work with clinical data on HIPAA de-identification. So HIPAA defines how we de-identify data, and we share de-identified data sets with investigators regularly. So if you want to identify all the patients with Parkinson's disease and their clinical histories for doing an observational research study, we can de-identify that data set under HIPAA and give it to you. But that data set is still risky. Actually, re-identification of an individual from that data set, you're not supposed to do it, but it is possible from a technical perspective, and actually probably not that hard. And so we're very interested in both creating walled gardens for use of data like virtual desktops. If you're part of the UW Medicine workforce, you've used them, or these so-called synthetic data sets that are basically fake patients that look like UW Medicine patients, but don't have HIPAA attached to them. We're huge fans of these emerging standards for how we structure electronic health record data for use for analytics and for research. One of the most popular of these models is called OMOP. It's sort of an esoteric name, but basically what it is a relational database that I'm showing on the right that can take visit-centric, patient-centric clinical records. So it's visit-based. So it's a patient with a list of all their visits, what happened in those visits, and what the outcomes were, if they're available. Very easy to build, enables exploration and learning, et cetera. And we will take that data model, and we can then, in the same way that that this person does not exist was created, we can create a synthetic data set of patients, and that synthetic data set, again, no HIPAA, is they look like UW Medicine patients, but they're not real individuals. So again, the barrier to sharing that data is much, much lower, and we think that the data is still useful for training, research, et cetera, and potentially even training machine learning in the future, like building AI methods. We're a little skeptical of that. And in fact, we published a paper just recently in Nature Communications with Brad Malin's group at Vanderbilt, where we proposed a model for taking synthetic data sets and evaluating the trade-off between privacy, that is, not being able to identify individuals within, like, say, the UW Medicine data set, and how useful they are for doing things like training machine learning models or doing other kinds of analysis that people are proposing that we should be able to do. And we built from Vanderbilt's data and from UW Medicine data, we built a number of synthetic data sets, and then basically had a battery of tests that we applied to them, like how well can they, how accurate are machine learning methods that are built on the synthetic data and then applied to the real data, and vice versa. And so you can read that paper. I think it's something I'm really, really proud of, and really, helps lower and reduce the risk, but not eliminate it completely from, clinical private data sets. We've done fun things with our data too. So if we look at all the patients at UW Medicine that UW Medicine has seen over the last 30 years and plot them on the state of Washington, you can see they mostly circle in Seattle. And if you zoom in on Seattle, maybe not surprisingly, the highest populations are near our hospitals of UW-MC, Harborview, and Northwest. Valley data was not included in this analysis. And one of the obvious questions that you might want to ask is, if we have this great data set, is like, how well can this data set tell us about population health, and how applicable is it to, say, the region of Seattle or to individual zip codes that are being shown here? And we've done lots of fun things. I'm not going to talk in too much detail. I'm just going to kind of like give you a really super high level of some of the kind of analysis that we did. So we took all of the diagnoses from our emergency rooms, and we then looked at those diagnoses on a tree for ICD and the International Classification of Diseases, which is how we diagnose people. You've seen them before. And we basically, for every term or every diagnosis within the tree, we put a circle, and the circle's diameter was basically how statistically significant it was for the region of time that we're looking at here compared to other times. And we looked at kind of different things like seasons, days of the week, and days of the year, that sort of thing. And what I'm showing you here is the ICD-9 tree for all of our emergency room diagnoses for the summer, and compared to the other 3 seasons. And what you can see is that it makes a lot of sense. You've got your water boating accidents, you've got your bicycle accidents, you've got your fireworks accidents, you've got your bites from insects accidents, you've got your snake bites, you've got your burns, all the things you would expect to see in summer are here, when you compare it to the rest of the year, because in Seattle, that's what we're doing in summer, and we're not doing it during the other times. We also did things like looked at kind of like what are the most significant, diagnoses that happen on certain days. Again, just showing that, doing big data kind of analysis from our data sets is actually relatively easy today, and it makes a lot of sense from a functional or causative perspective. So if we look at July 4th, not surprisingly, it's fireworks and alcohol and water boats, like and accidents. Like that's what is enriched on those days. And again, that's not surprising. We also looked at every day of the year, and the reason I kind of want to show this analysis is a lot of interest in healthcare and climate change right now, and there's a lot of RFAs in the federal government to fund these kinds of projects. And we looked at the most significant days for diagnosis codes for the emergency rooms over the past 30 years. And of course, a number of the days are holidays, because certain holidays always occur on the same day. And so they show up as being significant, and certain things tend to happen on certain holidays. Like on January 1st, people get drunk and they get in fights and they end up in the emergency room. We can see that from the diagnosis codes, and it comes up as significant. I already mentioned July 4th and 5th. People are, giving themselves trauma from fireworks accidents, and hitting other people on boats or something, and getting injured and ending up in our emergency rooms. But it's interesting, two of the most significant days of those 30 years that show up, one is the earthquake, the Nisqually earthquake, which was very surprising to us. And the other one, which is top 6, is the Hanukkah Eve windstorm, which is a climate event, a severe weather event. And so I think this is kind of an intriguing hypothesis to maybe look at more, in that not only might we look at smoke or pollen or other things that are associated with climate change and human health over time, like asthma or allergies or something like that, we could also look at how extreme weather events, if extreme weather events are changing, might be affecting other parts of clinical care, like say an emergency room. So if you're looking for, grants to write in this area, this could be an interesting thing to do. Epic is now using machine learning. All of the EHR vendors are. Cerner has dashboards, Epic has individual tools, and they're actual real machine learning, and they're giving them to sites to implement, and analytics teams are actively implementing them nationally and internationally as well. And they're based on the patient data population from that health site. We have a number of EHR models with our vendor that Epic provides. They're business models, inpatient models, ambulatory outpatient models. There's about 25 or 30 of them that we can evaluate. Things like, is a patient going to miss a clinic visit appointment, and do we need to do something to get them there? We want to look at inpatient deterioration or risk of sepsis, so we can alert early and treat the patient before it becomes clinically obvious that they're having a life-threatening condition. We can look at opioid use disorder, et cetera. And, the obvious question is, Epic gives us these models. We're smart people and part of a university. How do we govern implementation of those models? How do we measure the value of those methods? How do we ensure that those models are fair to all of our populations? I think these are very important questions that we need to have in place procedures or pathways to be able to get toward. Working with Beth Britt and a number of others, I sit on basically an AI governance committee that sits within IT, that's under the umbrella of our of basically our use of data within our executive use of data within UW Medicine. And this is somewhat complicated, but basically we've created a workflow by which a proposed model that could be either an Epic model or a custom model that was developed, like say by an anesthesiologist who wants to dose better in the OR, we are then walking through this process with them. And that process would be both the governance for it would be both contributed by a clinical leader as well as the IT and data leader. So it would be basically a partnership between analytics and between a clinical team that wants to implement that, and would require demonstrating the idea, validating the model on data, piloting the model, and then fully implementing the model, and then having an out phase where we watch the model over time. So here's an example of using some real data. So about 20% of patients miss scheduled appointments nationally. Looking at our data, and you can look at the data very different ways, but about 14% of UW Medicine patients miss visits, and you can intervene, right? You can spend resources to ensure a patient makes a visit. You could text them. We probably do that already. You could call them. You could send a taxi cab to pick them up. But you could also do what airlines do, is that you could overbook a clinic based on whether you think a lot of patients are going to miss that. And, one thing that I would caution is that there's a real worry that an intervention like, say, overbooking might harm one population over another based on the accuracy of that method. And sure enough, if we look at it, the methods are different the method is different in terms of its accuracy if we look at patients based on their annotated race, like Black, Asian, or white. And you could make a very strong argument that intervening with an overbooking of clinic visits would harm one population based on their annotated race over another, because the model is more or less accurate on that. You don't know whether this actually has a major effect in practice. That's like where we're trying to aim to go, but I think you get the point. So that leads to another question, is like, how accurate are these machine learning models really? And typically what we do is self-assessment. And I just did self-assessment twice in this talk. That is basically me evaluating my own method and telling you how well it works. And that approach for doing assessment of a model is rarely trustworthy. And implementation of them, of these models based without that self-assessment is really, really hard. That is, we know that that when people evaluate themselves, they tend to overestimate how good they're doing. They say they're doing better than they're really doing. We call that a self-assessment bias. And in the healthcare world, where we're dealing with protected health information, artificial intelligence methods are very, very hard to get away from that self-assessment. And I have a feeling that we're probably going to have groups that are looking at their models, and they're basically going to be thinking that their models are much better than they really are in practice. So I'm just going to give a quick aside. The University of Washington has recently, a little over a year ago, has blessed a new institute that is joint with the College of Engineering, the School of Public Health, and the School of Medicine and UW Medicine. It's called the Institute for Medical Data Science. And our overarching goal of this institute is to implement AI and data science into healthcare and to study it in practice, and to try to get us toward, to put the platforms in place to get us toward where I just described that we want to go. We have a number of aims. So we want to develop, open data sets that people can use to build models. We want to be able to develop novel methods. We want to be able to implement those AI methods into care. We want to study the downstream effects of those methods. And we want to investigate new biomarkers and data types that might improve care. And of course, we want to improve patient health and provider satisfaction regionally using AI. And we've defined a couple of different programmatic areas: clinical care delivery transformation, technology innovation, business and healthcare operations, societal impacts, advanced diagnostics, and education. So if you're interested, send an email to imds@uw.edu, and we can add us to the mailing list, and you can get involved. And there's a lot of opportunities now, a year later, to be involved with our growing institute. I'm going to skip that. All right. A couple of things. One, the Institute is running a workshop at the end of February on the 27th and 28th. It's almost free, so there's no reason not to attend if you have any interest in this area at all. You can use the bit.ly to get there. We're looking for posters and abstracts for short talks. If you want to do a short talk, they'll be competitively selected. You need to submit before the 1st of February, because we're going to start selecting them on the 1st, so that we have a good agenda for the meeting. It's going to be a great meeting. And we will have, the first day will be a half day, and it'll be an overview of medical data science with really, world leaders from the Seattle region telling us about different areas like FDA regulation of machine learning, the bioethics of machine learning, introduction to digital health, how to access and text and mine text, the development of advanced diagnostics, how we can use UW medicine systems to implement AI, et cetera. The keynote is Atul Butte, who I've known for decades, is really awesome. He leads the UCSF Center and is really the superstar of our field. And a number of UW centers and VIP faculty will be giving presentations that we know about that have been invited, including the Clear Center for Musculoskeletal Research, Jerry Jarvik, Shwetak Patel in the Allen School, David Beck, the director of the lead in the Science Institute, and Cecilia Lee, who's a co-PI of a new Bridge to AI Center that was founded by the NIH here at the UW. The other thing I want to mention about our institute is we also have a call for pilot projects that would be competitively funded. They're not big. I'm sorry. I wish we had more funds. Someday maybe we will, but it'll be $40,000 for a year with up to $20,000 in cloud credits for a total of $60,000. It requires having 2 PIs, one being the contact PI between computational and clinical faculty. So you have to be UW, you have to be UW PI eligible, and you have to bridge the computational and the clinical, but we really welcome trainees who have 2 mentors to apply for this, because I think it's a perfect mechanism for trainees. You can read the RFA at that bit.ly that's there on the screen. All right, I'm going to finish in the last 5 minutes talking about something I'm going to rush through, but it's something I'm really excited about. And that is, we want to take all of these cool things that we've done, and we want to build an open platform at UW Medicine for building novel machine learning or artificial intelligence technologies on UW Medicine clinical data. And we want to enable individuals from both inside the UW Medicine workforce as well as outside the UW Medicine workforce to be able to build those models using our data. And the mechanism I'm going to use for this, I'm not going to talk too much about this, is these sort of open experiments that you've probably heard about, like if you've ever heard of Kaggle or the Dream Challenges or the critical assessments or something like that, where we ask questions that data scientists can answer and then they can answer them. And we'd like to do that sort of an approach to UW Medicine clinical data to get a better insight about how well ML models are going to work on UW Medicine technologies and patients and providers. So we developed a really ambitious experiment. We've got a couple of papers on this. There's a few posters that are online that you can read with Justin Ganay at Sage Bionetworks and a number of scientists who worked with us, including folks from my group. And basically what we did was we took synthetic data. I told you about synthetic data in the OMOP data model, and we put it in the cloud in AWS. And what we did was we created a mechanism by which we could basically have an honest broker or an app store of Dockerized, containerized model, machine learning models that we could then apply to clinical data. And we created a mechanism by which a data scientist can basically upload their model into the cloud and provide instructions for how that model is going to operate when it sees the clinical data in the cloud, in this case, the synthetic data for real. But what we did was we also created essentially a backdoor where we were able to pull down the models ourselves and apply them to UW Medicine data that's real data, under an IRB, and again, with executive approval from UW Medicine to basically study how well these models being put into the cloud are accurate on UW Medicine data. I'm going to skip that. There's paper, Stanford, UCSF, Google published a paper looking at deep learning of electronic health record or clinical data, and showed that they could get a relatively good AUC of predicting all-cause mortality of a patient population from their clinical data. We thought that was maybe a great first approach for our project. And so what we did was we designed basically an open experiment using that platform I just described, where data scientists could predict 180-day all-cause mortality. That is, they could predict which patients are going to pass away and why within 180 days. And what we did was we collected models. We provided a clinical training set for them of about 1.2 million patients. Again, they never had access to that data. It was all done by us pulling the models down ourselves and applying them, but they could train on 1.2 million patients. And we then had basically a way of assessing the model in practice was a leave out set of about 20,000 patients. And then what we did was we waited a period of time, and then we stopped the challenge. And then what we did was waited 180 days and asked which of the models that were submitted best predicted the future at UW Medicine. We did this a couple of years ago. We had more than 375 data scientists participate in this experiment from all over the world. There were somewhere between 20 and 30 teams that were able to build robust models with this approach. And this gave us ROC curves and precision recall curves of the accuracy of these models. The best model had a precision and recall of about 0.5 that says if it predicts a patient's going to pass away, it's right 50% of the time, and it captured about 50% of the patients, which is kind of scary if you think about it. The best model was the University of Wisconsin's biostats group that submitted a very fancy gradient boosting decision tree model based model Again, this was a triumph in my view for open science because all of the models untrained were made available on the web through GitHub, and you can just download them and you can apply them to your own data. And we also got a scientific description of the under the hood of those models. We learned some things too. So for example, accurate models may differ widely in terms of how accurate they are on individual populations. So for example, if we look at annotation of race, once again, two accurate models that both have very good signal, AUCs of 0.95 or even better, and we then break down by population, by race, gender, clinical history, other things, we find that those models differ in their accuracy with respect to those populations, which suggests that we should look at, from a social equity perspective, we need to look at the accuracy of individual machine learning or artificial intelligence approaches on certain populations as they're being implemented. And I think this is really important. We did a COVID-19 challenge. We published this in JAMA Network Open. We ran 1,000 models over a little over a year, I believe. And we asked data scientists to predict whether a patient was going to test positive, and then their risk, once they tested positive, what's their risk of being hospitalized? Again, using UW Medicine data, clinical EHR data from that data set, the data sets that I've described earlier in this talk. I'm going to skip that because I'm running out of time. One of the things that we learned about it, so here's 4 models being shown over 3 periods of time, which are the box plots of different colors. The models are kind of descending 1, 2, 3, and 4. And the 3 colors, blue, orange, and green, are time period 1, time period 2, time period 3. They're approximately about 3 months, I think. What we see is that some models are very robust over time, and other models appear to be overfit. So for example, this model, which has an AUC on the first question of between 0.65 and 0.7, seems to be reasonably robust over time. It gets better as time goes on, whereas another model, this model shown here, started very good, and very good compared to the other models, and then in future data sets that happen in the future, the accuracy dropped significantly, suggesting that this model was overfit to begin with. And it really emphasizes how it's important for us to evaluate those models in practice. So I think what we've learned, the idea that electronic health record data is unuseful for prediction and data science is a myth. I think data sharing is not always required to develop accurate models. We didn't share data here. And machine learning algorithms can perform differently on different subpopulations, even if they are solving the same problem, trained on the same data, and are similarly accurate on the population. And even with rigorous multi-team independent assessment, overfitting of models, that is possible without continuous evaluation, and models will degrade in accuracy over time. We're continuing to work on this. We're working on a dementia challenge, working with the Memory and Brain Wellness Clinic and some others. And in fact, we've extended LEAF to be able to have patient-centric, population-informed dashboards. I don't have time to talk about it today, and we'd like to implement that into EPIC. I'm not going to talk about the National Alzheimer's Coordinating Center. We have a cloud platform that we've built for NACC, the Alzheimer's Center. If you're interested in advanced data platforms, my team would love to work with you and help you build that platform, assuming you have the resources to do it, which can sometimes be non-trivial. So I'm going to finish there right at 2:30. And I want to thank the research IT team for all the work they do operationally. I want to thank the National Alzheimer's Coordinating Center for all the work they do with Alzheimer's disease. And I want to thank my collaborators who work on the genetics projects, Peja Radivojac especially, and Vikas Pajever, both of whom I've worked with for decades now. Thank you very much.
Thank you for a really cool talk. Could you put up the next slide?
Oh, yeah, thank you.
Yeah, absolutely. So there are a couple questions in the chat here. So the first one would be Kaczynski writes, in using EPIC, there are often prompts that must be done to complete an activity. Sometimes I just click to close the prompt so I can get on with my work. This likely creates erroneous data. Similarly, when I cannot find an ICD code, I give up and find vaguely related options, again, creating unreliable data. Can you sort this out with your machine learning models?
That's a great question. So EHR data is notoriously messy. There's missing data. There's there's missing data, there's inaccurate data. Some of it's inaccurate for obvious reasons, like sometimes like a cancer screen might be annotated as cancer. And we see that, and that is going to affect the quality of a model that we see. That said, binary machine learning, like predicting whether you have cancer or not, is like, machine learning is like the great generalizer. So it's really good at like taking a whole load of messy data and turning it into something that's reasonably accurate. You could take an approach of curating individual patients, and I'm sure people are going to do that or already do that. So I think that's somewhat controversial in our area. We find actually that broader messy data sometimes is more accurate than curated data. And I could give a whole talk on that, but I don't have time. So I hope that's useful. I know that there's a lot of behavior within Epic that is tricky, that's going to result in weird data. And that would be something that we'd love to, maybe study better now that we actually are collecting that data. Looks like
Erica Johnlin writes, I'm aware that the FDA has guidance on the regulation of clinical data support software. I'm wondering how the FDA could regulate the use of AI to make the vast array of medical decisions and diagnoses possible. Could you comment on this? You mentioned that discussions with the FDA will be needed.
I'm not the world's expert on the FDA and AI, and there are people in our ecosystem that could help you. I think the short answer is if you start replacing doctor things that doctors do as part of their job with a computer, you're going to probably have to have a conversation about regulatory impacts of a method or tool that you're building. If you're just supporting a doctor, like building like a decision support tool, it's the barriers to doing that are much, much lower, because the doctor is still autonomous and making the decisions and has the agency to do that. I think there are tools like, so for example, radiology has implemented a model called AI Doc, which will triage images, medical imaging studies in radiology that are highly urgent, like brain bleeds or something that's like potentially life-threatening. And it's a deep learning approach that then will say, see an image come along and say, this image needs to be urgently reviewed because we think this is a very serious thing that has a time pressure to it. It will prioritize review of that, and that has been FDA reviewed and is approved for use for that purpose, because you're replacing a provider activity, which is selecting those images in the order to be reviewed.
Are there any other questions? I'll open it up for anybody. Just raise your hand or put it in the chat.
If you want to participate more in our institute, send an email to imds@uw.edu and just say, hey, I'd like to be added to your mailing list. And Nadej, who is our really awesome coordinator, who's been doing all this work, will make sure you get added. So I have a question. So I do a lot of sequencing and clinical sequencing, especially for tumors, is a rapidly growing and is already quite large. Could some of your models be used to go through like the variant list and kind of use that as prediction of response? Because right now people just like look at specific driver mutations or specific clinically relevant known ones, right? But could some of these approaches be used to kind of tease out like combinations of variants or something like that? Yeah,
or do you know of any initiatives that are working on it?
Yeah, so so our methods are developed for germline inherited variants. There are tools out there that are better at somatic variants than ours are to identify, say, drivers. The training set is different. So the training set going in would be, causative or likely causative pathogenic drivers that have been observed a lot. The group that I would probably point to that's done a lot of work in this space that I think is really awesome would be Rachel Karchin's groups at Johns Hopkins. And she's developed a number of methods that are like MutPred or Revel that are specifically tailored toward identifying driver variants in somatic cancer. So the short answer is yes, these types of tools can do this. The second one is my tools don't.
All right, we're a little over time, but I just want to, we have room or time for one more if anybody has a question. And if not, that's okay too. So want to thank you again for giving a talk. This is really cool stuff. It's tough to keep track of just how fast the technology moves.
We UW Medicine have a platform for implementing machine learning today, and I really hope people take advantage of that because it's an opportunity and it's, I think, a particular strength of this university now.
So yeah, hopefully people on this that were listening starting to get the wheels turning on how they can leverage these kind of resources. So thank you again for giving the talk. And then I believe in 2 weeks we have Ed Resnick at Sloan from Sloan Kettering giving the next R&D talk. So with that, thank you again, and I will see everybody in 2 weeks.